Chapter 7, Section 2, Transcription Factors and Effectors. Okay, transcription leads to messenger RNA. Messenger RNA is the instructions for how a ribosome should assemble the primary structure of a polypeptide. Well, how do you get the messenger RNA? You can't just have, RNA polymerases binding chromosomes willy-nilly. They have to have specific places to bind and start. So here, bind here, here's the beginning of the gene, and then go. And there's also got to be, a yes, no. Now's the time to do it. No, now's not the time to do that. That's where those things I kept alluding to in Section 1 come into play. These things referred to as transcription factors. They aid in saying yes, no, do it now or don't do it now. And then second thing they aid in doing is positioning RNA polymerase in the correct orientation and at the correct place so that any messenger RNA that will be made will have the complete gene or the complete operon. It doesn't start 10 nucleotides into the gene, 2 nucleotides into the gene, because then you completely mess up the codon reading frame of the ribosomes and you end up with some weird mutation, some weird misfolded polypeptide that has no shape and no function. As I mentioned in the last section, you have activator and repressor proteins. Activator proteins bind DNA, recruits in transcription, recruits in RNA polymerases or other factors, things like its sigma factors, to a promoter region. The more sigma factors in a promoter region, the more recruiting pressure on RNA polymerase. Because you got to remember, all the genes on a chromosome are in competition for a small number of RNA polymerases. So the more of these proteins can bind, the more of these promoter factors binding as a promoter, more of a me, let me jump the line that gene or that operon will have. The repressor proteins do the exact opposite. They bind, they mask promoters. They bind, interfere with other factors binding, sigma factors or anything else. Interfere with polymerase being able to bind and be positioned correctly. So when you look at what is happening with these transcription factors and effectors, and a lot of it's an allosteric interactions. So you have an activator protein in some form of an inducer. Maybe it's the presence of a sugar or a presence of, calcium, cyclic AMP, a metal, or a small polypeptide, whatever. Binds the activator protein. Activator protein is going to bind its site here, which puts it in a place to now recruit in other factors, sigma factors, other transcription factors, which will ultimately bring in and position RNA polymerase correctly at the promoter so that it can start moving down and making messenger RNA. A repressor does the exact opposite. A repressor gets activated by some co-receptor, some form of a sugar, metal ion, cyclic AMP, calcium, something. Binds the repressor. The repressor is going to bind here at the operator region. So basically, it's going to set up a roadblock so that even if RNA polymerase was able to bind at the promoter, as soon as the repressor starts moving down, so this repressor is going to bind right here. It's going to sit right there. When the polymerase moves down, it's going to hit right there, and it's going to stop, and then it's going to fall off. Boom. because this repressor that's bound to its co-repressor is going to have an affinity for that DNA sequence. Something about that sequence interacts very well with the R groups of the amino acids of this repressor protein that's now in the correct shape because it's bound to its co-receptor. So this, like I said, a roadblock. Whatever this co-repressor is, its levels drop in the cytoplasm, it will disassociate. It disassociates the repressor protein falls off. If any polymerase does bind on its own by happenstance, well, it can bind and move down along. So this is how these activators and these repressors work. Those effectors. Notice I was vague because there's, for every activator, for every, repressor, they're going to have their own effector molecules that bind to them and activate them. And that's going to be slightly different. Bacterial species to bacterial species is going to be different, prokaryote versus eukaryotic. We also have other allosteric proteins that when activated, their shape, that's what conformation means, their shape will change. In one shape, it has a function. In the other shape, it doesn't have a function. When it has a function, it's going to bind to DNA. Can act as an inducer itself. It binds, and recruits in polymerase. Does that bigger, ooh, me, I go next. Jump the line out of all the other genes and operons that need to be expressed at that moment and instant. Well, that's what an inducer does. Or these allosteric proteins, once activated, will act as co-repressors. They will aid those repressor proteins in setting up the roadblock, in interfering with polymerase binding to the double helix.